Today I'm going to talk about ChIP-seq data. So I'll talk a little bit of what ChIP-seq is, and then I'll go on to quality control of ChIP-seq data. Then I'll talk a little bit of big finding and mostly how the algorithm that we developed works. And finally, there will be like a short discussion. So the main idea of ChIP-seq is to identify protein interaction with DNA. And typically things look something like this. So you have if you're not used to 3D structure, so this is the one in field is a protein, and this would be DNA. And very often this protein will bind a specific sequence, and generally regulate transcription or do some other biological activity. And in the example there, it's OCT1, and which is known to recognize a DNA sequence, so it is GCIAT. And typically they look like this, so there are not that big protein and will bind like some small sequence of DNA between, it's typically between 6 and 13 base pairs. So how do we find out about these interactions? The way to do is chromatin immunoprecipitation, which is the ChIP part of ChIP-seq. So the first step is we do a cross-link with formaldehyde. So the protein is sit together on the DNA, and basically we fix it there, so it's not going to move. So now it's covalently bonded, so you cannot shake it off. Next thing we do is a fragmentation step. So we chop the DNA. And this can be done in two ways, either sonication, so you just a lot of sound on the machine, so this will just break the DNA, or we use some enzyme like MNase digestion. And the main idea is now we chop the DNA, so we have our protein is attached to this part of DNA, and the other ones are separated. And next thing is the immunoprecipitation. So we add an antibody that is specific to the protein we're interested in. And so at this point, it will attach to the protein, which is attached to the DNA. Then we wash everything, and we keep only everything attached to the antibody. So now at this point, we got rid of all DNA that is not attached to the protein. Okay, there is still some noise, but that's the main idea. And finally, now that we have our DNA and all proteins and antibodies, we release the cross-links and just purify the DNA. So we get basically all the DNA that was initially attached to proteins. And so this is the material of ChIP. Then afterwards, you want to find out what these sequences are. So either put it on a chip, which will be called ChIP-seq, or do next generation sequencing that will be ChIP-seq. And and that's pretty much the idea. So you just get this material, have it amplified, and get a lot of read. And so from, let's say, for example, from this initial sequence, you will have a lot of amplification, and you will have a lot of reads that go either this direction from here or this direction from here. So I will use the green for things mapping in the forward strand, and red for things mapping in the reverse strand. And and that's about it. So now we get a lot of reads, and they should come theoretically for places where the protein was attached to in the beginning. And so we do that. Then also very often we want to do control experiments. And during the control experiments, so we either omit the precipitation step, so we get pretty much pieces of DNA. So we get all the biases in the fragmentation that you get in the ChIP-seq. Or you can use a non-specific antibody like IgG, so that will just precipitate any protein. And so there are several challenges to do a relatively good ChIP-seq experiment. The first thing is to have good and specific antibodies. So you want an antibody that actually binds to the protein that you're interested in, and doesn't bind to other things you're not interested in. Which they're not always easy to find. And on the analysis, you have problem with the PCR amplification, so sometimes you will overduplicate, get a lot of artifacts, and I'll show a lot during this talk. And in the past, there was a big problem to get sufficient coverage. So very often you will get like very few reads. So you will get like, let's say 1 million reads, and that doesn't really cover the genome well. Nowadays, with the new sequencers, that's not really a big problem unless you have very little material. So how does ChIP-seq data looks like? So this is data from a transcription factor, and so we assume that is binding here and also here, but here much stronger binding and here a bit weaker. And so I have two replicates for an RSF is a transcription factor. So you will see that generally you will have a peak, and you will have very often reads that are mapping before the center of the peak that are green, so mapping in towards right, and red reads that will go in the other direction. And this is from the fact that when you get a fragment around, when you start sequencing, you will start sequencing from the left, and if you have something mapping in the reverse strand, it will start sequencing from the right. So you have a typical shift between the green and the red. And this is just to show, yeah. So the two replicates are quite similar, and then you still have a lot of read and control. And this is even those are higher, but not as high. And that could also be because when you do fragmentation, if the chromatin is open, it's easier to fragment that. So it's quite common that you get this kind of profile for controls. And that's also exactly why you do controls, because it's not like randomly uniform distributed reads. It follows some pattern of chromatin and sequence specificity. So next, the other big thing is histones. And histones are proteins that package DNA. And so it's typically, there are 4 units. And the important part is that you will have a DNA wrapped around it. And it's generally, the whole loop is about 147 base pair. And this one are basically very important in biology because they will pack the DNA, unfold the DNA. And for ChIP-seq, the important part is that the tails, so this is the tails of H3, are often modified. So you will find that some residues will have methylations or acetylation, ubiquitination, or phosphorylation, citrullination, or whatnot. And this different modification will be related to different stages of gene regulation. So some of them will pack the chromatin very tight, so nobody gets translated, or some will open it up, so genes will get translated, and more complicated things. And as I said, they are involved in many biological processes. So in chromatin remodeling and gene regulation, I put there only like 4 of the major ones. So H3K4 trimethylated. So this means you take histone 3, lysine 4, and you modify with the 3 methyl groups. And this is correlated to active promoters. So very often, if you see promoters, genes that are transcribed, you will have a peak of K4 trimethylated. K9 trimethylated, on the other hand, is repressed gene, and this is a constitutionally repressed gene. So there are genes that will never be expressed, and it's really tightly packed the chromatin. H3K27, it's also repressing, but this is more facultative. So when you have a cell, so certain cells will express only some genes, and generally those that are not expressed but are expressed in other cells are repressed by K27 trimethylation, while things that are never expressed, they're more packed together by the K9. And another one that is quite important, that I'm also going to talk about it a little bit more later, is K36 trimethylated. And this, you find it in gene bodies of transcribed genes. And so in this case, I'll show a little bit how the data looks like. So in this case, the peaks are wide and span large regions. So you can't just find like a single protein bind with the left and right, but you will have like a big region. And very often, if you're interested in gene regulation, of course, some of them will span the whole gene region. So there I have, for example, K27 acetylation. It generally starts quite high, then it fades down. K36, so it starts slow, it goes up. It's not so many reads here, but it's enough to say that it's actually present there. And as I say, K4 trimethylated. It's here the promoter. And I have a K9 acetylation that is also related to this gene being expressed. And here in the bottom I put RNA-seq data for the same cell line, and you can see that this gene is actually expressed. So because you have a lot of reads. So now I'll talk a little bit of quality control. So once you have a ChIP-seq experiment, what to look for, and to see whether the experiment went well or try to troubleshoot it. So the first thing that it's very important to do is actually have control. ChIP-seq experiments, as I was saying before, there are sequencing biases in fragmentation or in PCR amplification and other things. There are copy number variations, and so this is not only cancer, but for example, if you have a mitochondrial chromosome, you will get a lot of reads just because you have a lot of copies. So it's very useful to have something like that. And another thing is repetitive regions. So you will have microsatellites or retrotransposons, so pieces of DNA that are copying all over the DNA. So they will look like there are many copies of it. So you will get a lot of reads because just because there are several copies. And also, like centromeres and telomeres are extremely repetitive. So basically, anything mapping to there, we will often have a very high number of reads just mapping there, just because it's a repetitive region. So for those things, it's a good thing to have a control experiment, because then you will see a peak, very high peak in the control. And another control will be to do IgG, so you can correct for non-specific binding. And then there are some statistics that you can start to look into that. So first one will be duplicate ratio. So the duplicate ratio will be how many reads map exactly to the same place. And so it may be due to PCR artifacts or over-amplification. So if you do too many rounds of PCR, you will get a lot of the same thing over and over and over and over again. And you will get data that looks like this. So this is exactly what we don't want. This looks like PCR towers, and so none of that is biologically significant. So these are artifacts of the technology. And so a good metric for duplicate ratio is to look at non-redundant fraction. And a good threshold for that is 0.8. And I got these numbers from a paper by Modenko that is there in the bottom. So they have some guidelines. If you have a ChIP-seq experiment, try to look at these things and check whether your numbers make sense. Then an obvious one is sequencing depth. So you needed to have enough reads, of course. If you don't have enough reads, then you can't really sample the whole genome. And guidelines for these are, for transcription factors, will be about 10 million reads. And this counts uniquely mapped reads. And for histomarks, since you have bigger peak regions, they recommend twice as many reads, so 20 million. And nowadays, it's not really a big problem to get up to that number, so it's not much to worry about. And the next thing to do is cross-correlation analysis. Okay, so I'm gonna start from the beginning because I hope nobody has ever heard of this before, so I can take it slowly and not be too boring. So the idea of cross-correlation is that we have, as I was saying before, we have forward and reverse mapping reads. So forwards will be the green that I had before, and reverse will be the red. And the main point here is we want to align it. So since they are generally shifted, we want to find how big is the shift. And the most common way to do this is actually we just look at each starting position, each pair of starting positions. So we take one and one of the green and one of the red, and we just check, okay, which distance is more common. And in this example, I just simulated I have like one read here, two reads here in the same position, one here and one here, and just a little bit far apart. And the way we want to figure out is just if we count, okay, the distance between here and here is 3, distance between here and here is 3 as well, and this internal distance is 2, and this is 4. So we can just plot how many they are. And so, of course, and we can get an idea that shifting by 3 it's actually the right thing to do. And here on the left I show, so that's generally how in a transcription factor the coverage looks like. So you have this kind of shift. And this depends on very often on the fragmentation, as expected. And so it turns out that this activity is very well known mathematically, and it's called cross correlation. And I'll show a little bit an example how the thing works. So I'll put the formula in case somebody is interested, but the main idea is that you can have it with a star. So it's a cross correlation between F and G. So F will be the forward mapping rates, G will be the reverse mapping rates. And this is a function of tau, and tau will be the shift. And so the main idea, what we want to do is take, we have F, we have G, we start shifting G by 0, 1, 2, 3, 4, 5, and then we count, we take the dot product, so we say how much it overlaps. So if we start with 0, then there is no overlap, so 0 plus 0 plus 0 plus 0 plus 0 is 0, so 0. We take 1, same thing, no overlap, so 0 times 0 times 0 times 0 times 0 is 0. Now it's a bit more interesting. So we shift by 2, then this no overlap. This there is a 2 by 1, so 2, and no more overlap. So the basically now when we shift by 2, we actually get a 2, and if we shift by 3, then there's actually perfect alignment. So 1 plus 2, 3, and if we shift by 4, 1 plus 1 times 1, 1, and that's how we build again this cross correlation profile. And it turns out it's this is used a lot in signal processing because so it's very easy to do, and it's linear, and it's used a lot to find the lag between signals. So, oh yeah, that's a good idea. Oh, sorry. Okay. Oh, that's one. Okay, so it's used a lot in signal processing, and I mean, the most common thing is to find the lag between signals. So if you're waiting for some signal and you know that it's somewhere there, you want to find out where it starts. I know it looks like you just basically slide it and find where it best matches. So that's a typical use. And another thing is to match. So if I know that I'm looking for a certain shape, I can just run the shape that I'm looking for through all the signal, and this will tell me where it is. One of the nice things is that this is actually a relatively fast operation if you use Fourier transforms. So this is also used very much in like signal processing and a lot of things like that. Now, how do a cross correlation plot for ChIP-seq looks like? So there are several important features. So there will be a peak around the fragment length, and this is called phantom peak because it really shouldn't be there, but you always find it, and this is related to some artifacts. So generally, this is related to actually very often how bad the experiment is. So if this one is very high, you should start worrying. And so this is the whole cross correlation up to from 0 to 1500, and then here I just blow it up so we can see more clearly. And so that's the first feature that you should look for. Then the second is this high peak, and it should be about the fragment length. And this, of course, generally you don't know, so it's actually a good thing that you can do a cross-correlation analysis because you can figure out what the fragment length was in the beginning. And okay, so that's the other feature to look for, and the other one is background noise. So you will see that you will have, the more you go, you will have some background here. So here we stop at 1500, so this generally fades quite slowly. And this you get an idea, like if the background is very, very high, and this is not so much higher, it means that you have a lot of noise in the experiment. So clear enough. And so how to read it? You basically can just find the peaks, and then and get an idea. And there are some measures to actually characterize better what I just tried to say, which is so the first one is normalized trend coefficient. Which what it means, you just take the height of this peak and divide it by the background, and this should be, of course, high for ChIP-seq data. So in this example, it's 2.8, which is pretty good. So we can be happy with our ChIP-seq, and a good threshold for this is 1.05, which means this should be somewhat higher than this, because otherwise it's just noise, so we don't really gain anything by having done the experiment. The second thing to look for is a relative strength correlation. And so in this case, what we will do is we take this one, subtract the background, and this one, subtract the background, and take the ratio. So sorry, this should be minus. And so also in this case, you see this is higher than this, so it's 1.2, which is a good number. So it means actually that that we got specific signal, and this generally well correlated to transcription factor. And generally this is high for transcription factor because, as I say, the signal is concentrated, so you will always find a lot of reads around the binding site that have this kind of distance. So now I'll show what is bad. So if you see some cross-correlation plot that looks like this, you should start worrying. And there are two kinds that I find to be the main indicator of a bad ChIP-seq. One is if you don't have a peak at the fragment length, so it should be somewhere here, but it's really hard to see. And here you can see like the phantom peak is very high, and here you can see that this probably it's basically just noise. So the ChIP-seq didn't work very well. And another thing, if the profile is very jagged, so you see that it goes up and down very much, it means that you have very few reads. So it doesn't mean that they're necessarily bad, but it means just that you have very few. And that's just because if you don't have enough coverage, then basically this profile doesn't have time to converge, so you will have a lot of spikes just because you don't sample enough. If you had the same thing with a lot more reads, it will be much smoother. That's just to show that's not the only thing you can do with cross-correlation plots. You can, if you just look at the tails of the plot, you can basically distinguish some of the main feature of what the protein is actually doing. So in this picture, what we did was to take all the tails, all the profiles, and they're and we catalog by like different biological function that they have. And then we did a diffusion map, so we basically look at distances between the different profiles and try to cluster all them. And what will happen is, if you do this kind of activity, is that all the polymerases will go one way, all the transcription factor will go one way, and histone modification will go one way, and controls will sort of stay in the middle. And the nice thing about histones is that I was saying like they're wrapping around about 150 base pair. And you can actually see this pattern in the cross correlation. So you will have this, all of these peaks will be more or less one histone distance. So it will be a bit more than 150, so it will be about 170 because you have a linker. But you can really recognize that you have histones. And this example that is quite nice, it's K27 trimethylation. And this is one tightly packing, so actually you have a lot of histones next to each other, so you can really see the pattern very well. Okay, now I'll move to peak finding. So the main idea is not that hard. So we want to find regions with many reads, and that's pretty much all there is to it. The main problem, as I was saying before, is that there is a lot of artifacts and non-specific reads, and there are many different approaches. So I think, as far as I know, like about 30-40 algorithms, and so there are ways to model it. So you can, for example, model the reads that you have as a Poisson, or you can use like negative binomial, or you can use some type of clustering approaches. You can have a kernel density estimation, so you just basically slide a window and see how many reads you have in the window. You can look at something related to peak shapes. So in this case, there are some statistics on the shape. There are also ideas of taking the peaks and looking at specific DNA motifs. And another approach would be to use a hoteling filter, and I'm gonna talk about this later. And that's was used by the filter and also by the CLC one. So now I'm gonna start talking about the CLC peak color. And so our idea was to model the peak shape. And the main rationale for it is that so the peak shapes are an enriched region, and they have well-conserved shapes, as we showed before in the picture. And many peaks are clearly visible. So if you run any peak color, it will always find the top peaks because they are very obvious. And so the idea here will be to use a match filter. And the idea of the match filter is we know the shape it should have, and you just run it through the genome and find where the shape occurs. And the idea is that this is very general. It's been used in signal processing for many years. It's still specific because you can actually learn the shape that you're interested in, and you don't have to hard code it or just make up a simplified model. And one other big advantage that is quite intuitive, so you know that you're playing with the shape, and modular in the sense that you can learn the shape, apply the shape, so you can maybe tweak how you learn the shape or tweak how you apply the shape. And it's used a lot in signal processing, so it's quite stable, and you know what's going on, so it's quite well studied. And so the overview of what we're going to do with the algorithm will be so first thing, you just find a bunch of positive examples, so things that we want. And that's a typical transcription factor shape. So this will be in the experiment, you have a lot of reads, and this nice shape, like forward mapping in front, reverse mapping on the back. And the one in the bottom is the control. And so the control, we don't want a lot of reads. And this, so we basically an algorithm here will do, we just use a simple Gaussian filter. So we just run it through. When we find a lot of reads that follow more or less this shape, we just take it as a good positive. So now we have things that we like. The next thing is things we don't like, and this will be negative examples. So this is a typical thing that we don't want, and that's pretty much why we do controls. This is a macrosatellite, and what will happen that you will have a lot of read mapping here because this sequence occurs thousands of times in the genome, so pretty much it gets very much amplified. And so in the experiment you have a lot of reads, but in the control you actually have even more. And this is actually looks like it's something that we don't want, even though you have a lot of reads. And also you can see here it's not that clear that you have this nice shape. And this one also, to find it, just simply go through the genome and look for places that sort of looks like a pig, but have a extremely high control reads in the control. And now, once we have that, so we have a bunch of things that look like this, a bunch of things that look like this, then we want to learn a shape, and which we call hoteling filter. I'll describe it a bit more in detail later. But the main idea, we want to find a shape that can discriminate between these two. And so the main idea, we will have that, so then again, the shape is like reads mapping before, they should be high in the experiment. Reads mapping afterward will be high after the center of the peak, and then you will see controls they're actually they're less than 0. So if you have a lot of control, it will count actually downwards. So you don't want a lot of reads. And this shape is just learned from having a lot of positive and a lot of negatives. And once we have this shape, you can just basically look for it in the genome and just scan the genome and find things that look like this. Then again, how do we actually apply a shape filter to sequencing data? So here is again more or less how the average peak profile looks like empirically. So this is like a simplified filter, so I was just saying just a Gaussian filter. So what we do here, we get the fragment length from the cross correlation, like we saw before, and then you get some standard deviation as well from the cross correlation plot. You just see how wide it is. And the way you apply it is basically the cross correlation thing that we're looking from before. So you take all the reads, you look at the starting position. So if the read is mapping forward, then you have like a little spike. Green. If it's mapping in the reverse trend, we have a little spike in the red. And then if we want to calculate the value for this position, we basically take this shape, apply it here, and take the shape, put it here, and then we do dot product. So things that will, so this one will count quite a lot, and this will count very little. So we actually want a lot of reads, and we want them to be concentrated around here. And the same one for the reverse mapping reads. We want a lot of reads, and we want them to be concentrated here. And this we calculate for every point. And and for every point we get something that is a score, which is basically the dot product between the coverage and the shape. And typically on a transcription factor will look something like this. So it will go up and find a maximum pretty much halfway between the concentration of the forward and the reverse mapping. And it generally will look something like this. So it will go up and down. And also will be not too spiky, because you never know exactly where the center is, so it could be a few base pair before or afterwards. Then again, back to how we actually select the positive and negatives. So we ran the filter, so we had a lot of positive that hopefully look like this, and a lot of negatives that look like this. Next thing we do is to find outlier, and once we find outliers, we remove them. And so for this we use a Malanobi distance, and the idea of a Malanobi distance is that you take a distance between a point and a cluster. So what you want to do is not a distance between, let's say, a shape and another shape, but a shape and a group of shapes. And so what all this function means, that basically you look at the distances between the shape and the shape, but you also want to remove a correlation between points. And the main idea is that, let's say, for example, points that are here at the edge, they are really not that important, so you don't want to count them as much as things that are in the middle. And so that's the main idea. So we actually stretch things that are more important and remove and give less important to things that are not so much. And also at the same time we can check the distance between the shape and a cluster of shapes. So that's a nice thing about using Malanobis. And what we do is we calculate the Malanobis between any shape and so all the positives and shapes that are negatives and all the negatives. And what we find, that things that are very far apart we remove. And so for example, if we take so this pattern is showing like this cluster are all these things that were selected as positives, and this cluster is all the things that were selected as negative. And then we can calculate the distance from so for all the positive, distance from the positive examples, and from all the negatives, distance from the negative examples. And then we see that things that are very close to how a positive should look like have this very, very nice shape that we're looking for. But you can also select a positive that looks like this, which is kind of exactly halfway right, but it misses all the forward mapping grids. And this is actually a mapping artifact, because most likely this sequence is also somewhere else in the genome, so when you map it, it will actually map to the other place. And so even though this kind of looks exactly halfway like a positive, and it sort of will match because this half looks very much like the shape, it's not something we want to use for learning, because it's a bit messy. And similarly, like for negatives, we will see like that things that have a small distance from the cluster of negatives will look like the things we're looking for, while you can have stuff like this, which is just PCR artifacts. And in this case, you will have a lot of reads just because they just stack, and this is because they are repetitive region or PCR artifacts. And also these things, you don't really want to use them for anything because they're really spiky and really bad, and of course they're not representative of anything, just something that happened in the PCR. So a good way to learn, actually a good filter, is to get rid of things that we don't want. Now finally we are ready to actually learn the shape that we were interested in. And so a way to learn a shape, it's called the Hotelling observer. And what it will do, I'll try to go through the formula on using pictures and try to explain why every term is there. So the main idea you want to do is something to distinguish between positive and negative. Sorry, this should be P. So we want something that looks like this minus this. And that's the main idea. Then the other thing that we want to do is to remove the correlation between the points. So very obviously, let's say if I have a lot of reads in the middle, I will have a lot of reads just next to it. And that's sort of not a nice thing, so we want to remove this correlation. And the way to do is just to calculate the correlation matrix. And so we take an average of the positive and negatives and just invert it. And this can be trained from pretty much any set of positive and negative. So it's just as long as you can find some shapes and you can put it there and try to learn the filter. It has the nice property that it maximizes the OCROC. So it's the among all the linear filter that all the things that are linear that you can do, this is the one that will distinguish the best between positive and negatives. And one thing that is very nice for this is that you can have multi-dimensional input. So you can have, so in this case we separated forward and reverse and control forward and reverse. So we have like 4-dimensional input, and we could have replicates, or we could have like related factors and everything. And so that's a nice thing. You can just paste things together. So it's quite easy to do replicates and other things. And so once we run the filter again, we will get a score, and the score will be like we'll have a single nucleotide precision. So for every genomic position, we can say how likely it is to be the center of a binding event. And this is like, as I said before, like for every point, we do the cross correlation thing, so we can find out like how likely it is. And next thing that you may want to do is to, once you have a score, how do we get a peak? And so the way to go, or at least the way that we use, is to start from the center, and then we just expand left, expand right, and then basically so we go all the way here, and then we say like from this level how far do we go on this other side, and then basically we find we basically stop here because this one starts going up again. So, and then once we have this level, we just cut this level and then take everything from here to here. So that's how we get a peak. And so next thing we were working on was to look at Histone ChIP-seq data. And as we were saying before, for histones, peaks are wide and they do span a large region. So for example, in this case, it was gene. So we can't really do that same peak trick because they will get like a lot of very small peaks, about 150 to 100 base pair. So we wanted to use exactly the same approach, but just tweak it for having large shapes. And the nice idea also here that we can take every gene and just look at the coverage shape. So look at how much is the coverage in the beginning, in the middle, and so far and so forth. And so we can just extract the shape for every gene, and then, of course, if a gene is in the reverse strand, we will invert the shape so they match the other ones. And then again, positive will be just genes with the highest coverage. Negatives will be genes with the lowest coverage, or just get some profile of how the noise looks like. Then again, we build a filter and match shape with the filter. And this one, instead of finding like base pair by base pair for each gene, we actually get a single score. And for example, something that from the data before, we could see, like if we take like 2 factors, we will see, for example, that K36 starts quite slow and goes up, and K27 like starts high and goes low. So we can build a filter like that and just say, when we look at the coverage profile of the gene, does it look something like this or not, and how much it does. So now I'll talk a little bit on how you can evaluate performances for a ChIP-seq. And so this case, we use some manually annotated transcription factor binding sites. And so what they did in this paper was to go through a data set, run some peak color, find some peaks, look at them, and say which one were good, which one were bad. And so in this case, I'm looking at a transcription factor called max. And so here I have like the experiment control. And so if you run the algorithm, we will get this sort of nice peak shape score, and it will find that the center of the peak should be somewhere around here. Now, how do we validate things? So generally, you have some regions that are good, so positive, and some regions that are bad, negative. So in this case, they wanted to say, okay, you should find a peak that is here, but it should not have flanking regions. And so the way you can do if you have something that is continuous is just basically you take the region and take the highest value in the region. So for this positive will be here, for this negative will be somewhere here. And then again, if you use the normal peak color, you just look at the peak and get the value from there. And this is just indicative to say, for example, what happens if you use different replicates, different controls, and how much the performance change. And so in this case, not so much. So even if you run it with like 2 replicates, and for the chip and 2 replicates for the controls, you get pretty high Ocrox. But so if you start, for example, if you remove the control, it gets worse, because it's not that easy to learn, and you will get some artifacts, some of those with a lot of reads in both places, and you will see that it goes down, but not very much. So, and that's because transcription factor shape is very clear. So you can actually, if you don't have a control, it's still okay. Although it's much better to have it, of course. And another thing that is interesting to look from here is that if you merge the reads from the replicates, the results are slightly worse than actually consider them separate. So in case you have replicates, do not merge them before running a ChIP-seq in general. And also most peak colors nowadays, you can specify multiple inputs. So that's also something to keep in mind. And yeah, and this one is just showing, like if you take like only 1 replicate, so results are about the same. So it could even be lucky and be slightly better, but that's just a statistical fluctuation. And yeah, and this just to show that basically, so we use a bunch of algorithms to test, and so the results are quite similar. And so we tested like the CLC. So this will be the score, the one that was every base pair. This will be the peaks that we call, and this is cisgenome. this is cisgenome, but refined. So what it will do, it take the peak and find smaller regions that that are supposed to be the binding sites. Then we also run Homer and Max. So I would say basically there is not a very big difference if you run a peak caller. The advantage that we have is that if you run the score, since it's a base pair resolution, you actually can get a higher whole clock just because you call every peak. So that looks slightly higher, but I mean it doesn't really affect you unless you want to find very weak binding. So that's about it. And so the next thing that we did is to look at histone data. And so to look at this, it's a bit harder because we didn't have a good data set. So what we did is to look at transcription factors that histone modification marks that we know that they are related to gene expression, and see whether if we find a big peak there, it would actually correspond to a gene that is expressed. So what we did is just run RNA-seq, find out which genes were expressed, and then rank the histone factor ChIP-seq, and then see which one will be expressed. And we ran those on 4 cell lines. And so actually, the nice thing is that H3K36, it's well known to be related to gene expression you find in the body. So actually here you get a pretty good OCROC. So the main idea, you can pretty much predict if some gene is going to be expressed just by looking at the ChIP-seq of H3K36. And these other ones are correlated, but they're a bit less correlated. So I think the problem is not really in the peak calling, but it's just because the relation between the two is not as strong. But you can still pretty well like get an idea of what's going to be expressed. And yeah, now I'll just wrap up. So what do you do once you do a peak calling? So you run peak calling, generally you get a bunch of peaks. So if you have a transcription factor, the thing to do is generally to look at binding motifs. And so the idea is, does the transcription factor recognize a sequence, and if so, which sequence is it? And the second thing that you want to do after you recognize the sequences is like, is this sequence common to any other transcription factor? And that's also very fast, because if you don't want to do a ChIP-seq of 100 of factors, you can just look at the sequence instead of finding running all the peak calling for all the factors and see how much of the region overlap. So this is also quite a fast analysis. Next thing you may want to do, like some more functional analysis. So just look at peaks, find which genes are nearby, and then characterize like, are these affected genes part of some pathway? Do they have some gene ontology term that is interesting, or is something particularly enriched? So that's also a good direction to go. And of course, then comparing is also something that is quite important. So you can look at combined effect of multiple transcription factor histone marks, or the common thing like different conditions. So if you have some treatment, you can look at what it looks like before, what it looks like afterward. If you do some knockouts to see what happened, and yeah, so far and so forth. And so now I'll talk a little bit on similar protocols, and because very often people will use ChIP-seq algorithm for some of those, and it's not necessarily a bad idea in most cases. So I'll just describe like a little bit some of the protocols that you can use it for. So there is a ChIP-seq that came up a few years ago, and the idea of ChIP-seq is that once you do the immunoprecipitation step in the ChIP-seq, so you will have a fragment, and you will use exonuclease on the fragment, so your fragment will become smaller. And then when you start sequencing it, since your fragment site is smaller, then you will be able to pinpoint where the binding site is. And there is a derivative of this called ChIP-nexus as well, but it's pretty much the same idea. Another thing that looks quite similar to ChIP-seq is MEDIP, and you just so instead of looking for DNA protein binding, you look for methylated DNA. So this is a way to find out roughly which areas are methylated. So you don't go to nucleotide resolution like bisulfite sequencing, but you can get an idea like which areas are methylated or which areas are not. And then there are a bunch of methods to identify accessible chromatin. So the most common one that I know of is DNase hypersensitivity. So you will basically cut the DNA with DNase 1 and see where you get it. So in this case, so if you use a ChIP-seq algorithm, we will basically find regions where you have a lot of those. Then there are also other analyses that you may want to do on it that are very specific to DNase. So I wouldn't stop at just using a ChIP-seq algorithm, but it's a good starting point. And then a similar technology is Ferri-seq as well. You will find some open chromatin regions. And a more recent one that is becoming more and more popular is ATAC-seq, and where you just use transposons and as well try to figure out which part of the chromatin are open. And this is becoming more and more popular because you need very little material and it's very fast. So they might take over at least this one or maybe also DNase. Right. So in case you want to use this software, so the PIC-shape base color is in the genomics workbench and all the other CLCbio software. So we'll have a hands-on later. I think we'll get an idea how it looks like and how to use it. And so there is just a tool for doing ChIP-seq, and there is also a plugin for where you can do some more advanced things. So you can learn a filter if you give your own positive and negatives, and you can reuse a filter to other data, so which is nicer for comparison. And the thing I talk about is on ChIP-seq will be soon available for the next version of genomics, which is coming like next week or the week after that, I think. And in case you're interested, there are tutorials and white papers on the Piccolo. And yeah, just people that worked on it. So it was me and then Michael Lappe and Jens work on some of the machine learning parts, then on visualization and Morten on the histone modification stuff. Yeah, I think I'm done.